system in patients with severe stenosis of naval aortic valve. I don't have any conflict of interest. My valve is a novel balloon expandable transcatheter heart valve, the novel hybrid honeycomb cell design concept. 47% of the lower expanded valve frame has closed cells which provide high radial strength. Upper 53% of the expanded valve frame has open cells to ensure easy access to the coronary ostia. The frame is made up of nickel cobalt alloy, which provides high radial strength and good radiopacity. There's bovine pericardial trileaflet valve, an external PET buffing to minimize perivalvular leak, and an internal PET sealing cuff. Comes in 20 millimeter, 23 millimeter, 26 and 29 millimeter sizes, and these are the intermediate sizes in the pipeline. My valve transcatheter heart valve device is crimped directly onto the balloon. The crimped valve with delivery system is then loaded to a 14-16 French Python expandable sheet. The navigator balloon delivery system has a set of proximal and distal stoppers which ensure that valve crimping is precise and snug. Visual confirmation of the crimped valve can be done before entering the sheet. The proximal and distal balloon stoppers prevent inadvertent migration of the valve and ensure there is no risk of valve dislodgement during entry through the sheet or while negotiating the loaded delivery system across the aorta. My valve transcatheter heart valve crimping on balloon makes TAVR delivery simple, intuitive, and eliminates unwarranted procedural steps. The delivery catheter of my valve is called navigator transcatheter heart valve. It is a 14 French 120 centimeter long delivery catheter. The proximal shaft has a rotatory handle and a marker line which indicates the degree of flexion happening at the distal end of the catheter, thereby preventing scraping of the outer arch and facilitating difficult LV entry. My valve 1 study is first in man safety and efficacy study, enrolling 30 patients with severe symptomatic aortic stenosis. Clinical follow-up is 30 days, 6 months, and 1 year. Device sizes 20, 23, 26, and 29 millimeter valve. 14 investigating sites and India are involved. Echo Core Lab is situated at CRF and New York, USA. These are the researchers, Dr. Samin Sharma, Chairman, Ashok Seth, PI, Praveen Chandra, Coordinating PI, myself, Co-PI. 30 subjects all across the country were enrolled in the study. Key inclusion criteria was patients above age of 18 with severe symptomatic aortic stenosis and an STS score of more than or equal to 4. The exclusion criteria was acute MI within 1 month of treatment, bicuspid, unicuspid, or a non-calcified oblique rheumatic heart disease, mixed aortic valve disease with predominantly regurgitation, pre-existing prosthetic heart valve in any position, untreated significant CAD requiring revascularization, hemodynamic instability, EF of less than 20%, lady aortic annulus of less than 18 mm or more than 28 mm, renal insufficiency, patients on end-stage renal disease and dialysis, and significant aortic disease. These are the baseline key demographic characteristics. Average age was 75. 73% of the patients were male. Average STS score was 6.42. 80% of patients were in NYHA class 3, 20% in NYHA class 4. CAD was present in 43%, previous MI in 13%, previous CABG in 16%, previous PCI in 13%. None of the patients had balloon aortic valvuloplasty in past. One patient with a prior CVA but was not disabling. Peripheral vascular disease in 10%, COPD in 46%. 6% of patients had creatinine of more than 2. Baseline AF, atrial fibrillation in 20%. None of the patients had a baseline permanent pacemaker. Pulmonary hypertension in 16%. Porcelain aorta in 6%. And liver disease in 3%. Average perimeter-derived diameter was 23.9. Average area-derived diameter was 23.58. These are the average LVOT dimensions. Average RCA height was 12.92. Average LCA height was 13.63. Average sinus of valsalva diameter left 28.76, right 28.67, non 31.0. Out of the total valves implanted were 30. 20 mm valve was implanted in 6% of patients, 23 millimeter valve in 43%, 26 millimeter in 36%, and 29 millimeter in 13%. Pre-dilatation was done in 86.7% of patients. with balloons used were 16 mm, 20 mm, 23 mm, and 25 mm. 100% of cases were done from the transfemoral route. Right common femoral artery was used in 73% and left common femoral artery in 26%. 60% of patients had GA during the procedure and 40% were done under conscious sedation. These are the baseline post-procedure and 30-day echo parameters. Mean gradient baseline was 47, post-procedure 5.83, and at 30 days it remained at 6. Peak aortic valve gradient 71 at baseline, post-procedure 8, and at 30 days it remained at 8. Similarly, transaortic velocity 1.8 at 30 days, remained at 1.85 at post-procedure, remained at 1.85 at 30 days. EF remained the same. Moderate or severe mitral regurgitation was present in 2 patients, and it did not change at 30 days and at post-procedure. None of the patients had central aortic regurgitation. Mild paravalvular leak was seen post-procedure in 2 patients, and it remained mild at 30 days. None of the patients had moderate or severe paravalvular leak at 30 days and at post-procedure. Major adverse cardiac, cerebral, and renal event: 0 mortality due to study device. All-cause death was 1 due to kidney failure. Cardiovascular death 0. Repeat hospitalization within 2 weeks was in 3 patients. 1 was related to GI issues. 2 patients had vascular site access complication. 1 readmission at 30 days, again due to vascular access site complication. Stroke was not seen in any of the patients. 1 patient had renal failure, access site complication in 4, permanent pacemaker in 0 post-procedure and at 30 days, myocardial infarction 0 at post-procedure and at 30 days. Alternating V-fold and vertical connectors give my valve a unique appearance under fluoroscopy of alternating dense and light band, which help in precise positioning. Unique crimping geometry of my valve ensures precise orthotopic device placement without any deep throttling into the LVOT. Precise placement ensures there is no conduction system disturbance, thus eliminating need for new pacemaker as in with previous technologies. Re-intervention, post-procedure in 30 days, none of the patients required balloon valvuloplasty, repeat TAVR, aortic valve replacement. MyValve 1 clinical trial conclusions.
So I think you're a bit tight on time, so if you could just summarize maybe the conclusions.
MyValve 1 first in man has successfully completed the primary endpoint demonstrating high safety at 30 days, 100% device success, high clinical safety with 0% device-related death, high clinical efficacy, mean gradient post-procedure 5, which maintained at 30 days, peak gradient post-procedure 8, which maintained at 30 days, and the aortic velocities of 1.8 post-procedure, which maintained at 30 days. Improvement in NYHA class, 0% peri and post-procedure stroke, TI or MI, which sustained up to 30 days, and 0% new pacemaker implantation. Thank you very much.
Thank you very much indeed, and sorry for the tight timeline. But these are very impressive results, albeit in quite a small number of patients. Is there an ongoing follow-up study or access for a larger number of patients? And is it going to be restricted for use in India alone, or it will be more widely available?
So at present, definitely the study has been extended. We plan to enroll almost up to 70 more patients for the first in-man study. And the ongoing, the future plans are to definitely extend beyond India as well as extend the indications also. Okay. Thank you very much.
So we're very happy to take questions from the floor. There are microphones at positions around the room, so please do make your way to those if you have any questions. Raj.
So Ravindra, did you exclude bicuspid aortic valve stenosis?
Yes.
And the next question I have for you is, the pacemaker rates were 0, and do you really think that this is just a matter of chance because it is only 30 patients? Or do you truly believe that this is related to your positioning, and did you actually look into it? Were you really, what was your average depth, if that is what your theory is?
Yes, so I think both the points are very important. Definitely the number is 30. But what we have seen in all these 30 cases is because of the good radiopacity markers on the crimped valve frame, we were almost always 80% aortic and 20% ventricular in all